Hello, my name is Haris Nawaz, and your mama is so fat that when she sits on a chair, she sags out both ends. Now, when I first heard this, I was horrified. I ran away and I found myself at the health genetics lab studying polyploids. Now, what are polyploids? Well, you and I are diploid. We both have two sets of chromosomes, one from each parent. However, there are many organisms that are polyploid. They have multiple sets of chromosomes. They can be triploid or tetraploid, meaning they have 3 or 4 sets of chromosomes respectively. However, there is a polyploid version of you right next to you. It looks exactly the same. So, how does that relate to our experiment? What we're trying to do is create as many polyploid flies as we can, fruit flies, Drosophila melanogaster. We're trying to do that through something called cold-induced polyploidy, or just cold treatment, to stick them in the fridge and see what happens. But what's the point of that if we can't tell the difference? They look exactly the same. For the purpose of our experiment, we use something called genetic markers. For the basis of this, just try to understand that if we have both the drop and stubble genes, which you can see right here, in one fly, we've gone ahead and created a triploid, which can be reflected in our experimental design right there. The plus will indicate the wild type, and the drop and stubble genes are the phenotypes for the mutations. So if we have both of them cool in one, then we've got a polyploid. Going into the genetics of that, it's something called meiosis. You go ahead, you have that normal female cell, the diploid, drop and stubble, go ahead and separate, and then you go ahead and drop and stubble are going to be in different cells. You go ahead, you get them mated, you get some that are drop or stubble children, which is our normal room temperature, 25 degrees Celsius mating. However, you have some that are drop and stubble, non-disjunction can occur, which just means that the chromosomes don't separate properly. Cool, you get them in some of the same cells, and now you have some that are drop and stubble, get mated, now you have the kids are going to be triploid. Non-disjunction can occur on its own, although it's closer to 0% of the time, so not really. But however, we can use something called cold treatment, stick them in the fridge, and now go ahead, non-disjunction occur, and now these are going to be triploids. This is very cool and all, but going through hundreds of these flies and going through it all, our model still remains. Kind of fun when you're having flies. But moving on to the human impact. How is this important? Cancer cells, a lot of different types of cancers, are polyploid. This is very dangerous because a lot of these polyploid cancer cells are very resistant to a lot of different types of therapies. Better understanding how we can fight against these types of cancers through polyploid research can help us better eliminate these types of cancers. Another interesting phenomenon is called parthenogenesis, where certain species of flies are able to reproduce on their own, recruiting different egg cells and fertilizing themselves, creating polyploid offspring. Through CRISPR, which is just genetic editing, taking different chromosomes and working around with them, we can go ahead and maybe induce this in humans later on, which could help us fight against a lot of genetic diseases. Overall, though, this is very important because a lot of people see chromosomes as insignificant. A lot of these fruit flies are just tasty little snacks, which they are, but however, they're still very important because there's a constant reminder to me, especially, that many different types of symptoms are important in the medical field. And to wrap up, though, I'm very happy to share this all with you. And to wrap it up, I've gone ahead, went back to that person who called my mom fat, and said, she's not fat, she's just a polyploid. Thank you.